Hi, I'm Jeremy, founder of the RegenReport.com, where we cover the regenerative medicine industry. I think most people are aware that, modern medicine has brought us a lot of fantastic technology that's kept us on our feet for much longer than God intended. But unfortunately, there are still a lot of conditions that we just don't have answers for, especially in the sort of neurology area, and more specifically, spinal cord injury. However, some of these conditions are potentially going to be solved by regenerative medicine. There are a variety of new companies popping up with tech that looks pretty good. Most of it's in the preclinical or very, very early clinical trial stage. And today we're going to interview one of those companies. And the company is called Nurexone Biologic. They're a startup based in Israel. And before I get too far into what they do, let's just start off with the visual. So they do an exosome product called ExoPTEN, and it's used for spinal cord injury. It's in the preclinical stage, and let me just show you what they found out. So, on the left side, you have a rat with spinal cord injury, the control group. They did not treat this group. On the right side, you have the group that was treated with the ExoPTEN exosome product. And just take a look at the hind legs. You can really see a stark contrast between these two. On the left, essentially dragging in the untreated group, and on the right, it's regained a significant amount of its motor function. You can see that, especially again in those hind legs, basically dragging and pretty much walking on the right, which is really interesting. And ExoP10 is Nurexone Biologics' lead therapy candidate right now. Essentially, it is an exosome therapy where they take exosomes derived from mesenchymal stem cells, load them with siRNA, and that can help target a pretty wide variety of nerve repair. But they're starting off with acute spinal cord injury because they received what's called FDA Orphan Drug Designation. When we have a condition that we just don't have a lot of answers for, like spinal cord injury, if there's a therapy that looks pretty promising in the early stages, the FDA will give this special designation to hopefully get that therapy to the next stages a little bit faster and a little smoother. And they received that in 2023. And today we sat down with the CEO, Dr. Lior Shaltiel, who's going to break down what ExoP10 is, how it was made, all the science behind it, and what they found in those preclinical results, and where ExoP10 is going, hopefully in the next couple of years. If you don't mind liking and sharing liking, commenting, and sharing the video, it's how we get the regenerative message out to more people. And if you want to stay up to date on interviews like this, new trials, new research, new industry news in the regenerative medicine industry, I'll put a link below in the YouTube description for the weekly newsletter. You just pop your email in here and we'll start sending it out to you. So enjoy the interview. Could you start off with an introduction to you and Nurexone, a little history?
Absolutely. First of all, thank you for having me today. Happy to be here and to represent Nurexone, a relatively young startup company established in 2020, coming out from two elite university in Israel, the Tel Aviv University and the Technion, which is the MIT of Israel, located in Haifa. Two professors that joined forces during the last 3 years, and their focus is to regenerate neuron in the spinal cord injury. They tried different methods, and the best method was what the company is doing now: exosomes loaded with siRNA against the protein called PTEN, PTEN, in a minimal or non-invasive administration for spinal cord injury. That's the beginning of the story. The company was established, as I said, 2020. I joined as a CEO in April 2021 when the licensing agreement was signed. My responsibility was to take the company and establish the company, build the IP, the team, and the rest. That's a short background, a bit background on myself. I'm a chemical engineer as a bachelor, master in physiology, PhD from Germany, LMU, Ludwig Maximilian University in Munich, in pharmacology. I had a short postdoc in the University of Frankfurt, the Goethe University, and I returned to Israel as a return scientist back in 2014. Since then, I work in the industry, mainly in drug delivery system. I work with liposomes. Now we are doing exosomes. I have also academic background. I'm associate with the Hebrew University Business School, established their unique MBA program focused on biomedical management. This year we celebrate 10 years of this program with dozens of alumni, even hundreds. And yeah, that's my scope. Try to promote biomedical excellence and innovation into our companies. And in Nurexone, we do it to the extreme. A very interesting science, technology, and the fact that we are turned to be a publicly traded company in a very young stage. And we will speak about it today.
Awesome. And so it's ExoPTEN, right? That's what you're,
Yes.
and that's
ExoPTEN is the name of the product.
and that's your flagship product. Or that's most of what you're doing is ExoPTEN right now, right?
Correct. This is our first and lead product. What is unique about it is that this product, a product that is already proved in a preclinical in preclinical experiments in different models, spinal cord injury, two different models, optic nerve, and also facial nerves to be effective. Of course, it's a different concentration and different administration if you're talking intranasal, intrathecal, subretinal, or local injection, but in all cases, it's the same product.
Okay. Could you give a really quick overview of P10 and then we'll lead into what ExoPTEN is doing and how it's targeting that?
So PTEN is known in science for many years, say 20 or 30 years, mainly in the oncology is a key factor in the cells to keep the balance between proliferation and to be or not to proliferate. And this is why it's a tumor suppressor. It's part of the mTOR single transaction. Okay, and when you are downregulate this protein, you actually promote proliferation. So in another way, what we are trying to do is to downregulate or to inhibit the inhibitor. Okay, when you inhibit the inhibitor, you are pro-regenerative, you are promoting proliferation and exon growth. Okay, so of course, if you are downregulate in a very aggressive way, or even knock out the protein, is lethal or dangerous to create a cancer. But the fact that we are doing it at a very localized, temporarily, okay, is that the advantage is sometimes, everything that you do too much is bad, as a lot of people say. We are doing it a bit just to help the cells, help the body to get over the damage. And when you have a damage in the spinal cord injury, you have their inflammation first. And after the inflammation, you have a scar. When you have an inflammation, it's the opposite process that the body needs to have. When you have inflammation, you cannot have regeneration. So the idea is, first of all, to use the inflammation in order to accumulate exosomes because they're homing to the area of the injury, reduce the inflammation. They are very immunomodulator in our body as exosomes. And then they also promote regeneration in the certain cells, mainly the neurons, okay, in the area of the injury. So you have two function in one product, and we tick it, I would say, we add another synergistic effect by using the siRNA.
Awesome. And let me just back up a tiny bit. So I think most physicians and scientists know exosomes. They're extracellular vesicles secreted by cells, and they are different than putting stem cells into your body. Could you talk about some of the advantages of using exosomes as opposed to stem cells and how that sort of influenced how you developed the product?
Yeah. So also during the research in the university, it was quite clear for the researcher that stem cells, iPSCs, MSCs are very relevant to promote regeneration. But with the time, many researchers understood that the effect of the cells is actually the effect of what they secrete. And what they secrete is those exosomes. So that now we have another concept. We have a cell therapy without cells. So if we just inject the exosomes and we will see the same results, why we need to work as with the cell therapy? We need just what they secrete. And this is actually what we are trying to do. Take what they secrete, try to help it with another siRNA to give a synergistic effect and turn more to regeneration and neuron protection. And by that, in the acute phase of the spinal cord injury or acute phase of any central nerve system injuries, we can protect and protect the neurons and promote neuron regeneration, neuron rewiring. And what is also important is not that they just rewire, just connect. We want to see and measure the functional recovery, how much we can recover from what we lost due to the injury. So a lot of VC will see, if you're doing with spinal cord injury, I want to see a human or a rat that walk again. But motorical function is not the only things. We are talking about sensoric. I mean, do you feel? Do you suffer from pain, which is very, very common in spinal cord patients? Can you reduce phantom pain? Can you increase sensitivity? Can you suddenly control your rain bladder? Those are the things that are important for those patients, not only the mobility. Okay. To walk again is a very important goal, but it's not the most important.
Yeah, absolutely. I guess can we talk about some of the results? So you've
Yes.
applied this in rat models so far in preclinical, and can you talk about what you found out? How did you administer it? What did the rats have? So you spinal cord injury, and what were some of the results? Can you walk through these clinical trials? How's it working?
So we did it in two steps. In university, they use a proof of concept, a molecule that is not freedom to operate. When you're going to a company, it's very important. In general, when you are talking about innovation coming from academia, you need to understand if this product is freedom to operate. So the first step that I had to do as a company and instruct my employees is to make sure that all components of the product is with the freedom to operate. So in this case, the source of cells needs what is the source of cells? Okay, it's bone marrow mesenchymal. From where we take it? Okay, is it our donor? Is it we purchase it from another company? This is sorted by the fact that we bought just recently a master cell bank. Second, how you produce it? Do you produce it in 2D, 2 dimension, or in 3D, 3 dimensions? If you use 3 dimension, do you use a scaffold? Do you use a carrier? There are different techniques. We're using our own way to produce it. By increasing shear force on the cells, we can produce more exosomes, and they are more potent. And then we are talking about how you modify those exosomes. Our way is to modify the exosomes by loading them post-production. After you produce the naive exosomes, you can now inject them, so to say, or load them with the siRNA. This is how we do it, and this is also protected as a patent in the PCT stage. And the last thing is what you load inside the exosomes. Is the sequence to load to downregulate the PTEN protected? So in our case, yes, we protect the sequence, we protect the chemical modification to make it stable, okay? And in this case, this is the first thing. So is the same, maybe the same name, ExoPTEN, but the new product with the new components need to be tested again. So we tested this new component with the new product with two different models. The first one, we repeat what happened in the academy. In the academia, they use full transaction model. Your transaction model is not really common in industry because it's very it's non-physiological model. It's very aggressive. You take a rat, cut completely the spinal cord injury, and ask the question, can I rejoin the nerves that are damaged and show functional recovery? And indeed, we could do it again, and we saw again and again results with that. This was done in intranasal administration, okay? And we move also for intrathecal administration to make sure that we actually can maybe reduce the numbers, they reduce the concentration, reduce the numbers of the injections that we give during the therapeutic window. So that's the first model with two different administration. The second model, we said, okay, we passed the first step. We could see that we can repeat the results that the university show, which is sometimes is an issue. Statistically, you're talking about 50% chance to reproduce results. So we passed that. We have more confidence that we have something in our hands. Second, we're saying, okay, let's go to a physiological model. And in this case, we chose to go with compression. Okay, so you take the rat, press a certain level of the nerve, spinal cord. In this case, it's T89 in the back, and let it recover during a 2 months period when we inject once, or it was several times during the 2 weeks. Okay. And in the last results, you can see that a high dose of ExoPTEN really can cause 100% of the rats in this group to walk again.
100% in the group.
100%.
How many?
It's much easier. It's much easier if we saw 50 to 70% in the full transaction recovery. Now we see 100%.
How
And
many?
you can see
How
that the
many
dose
rats?
dependent. We are talking about dozens of threats already that we had operated in these different experiments. So we are comfortable, and then add to that, we said, okay, if it's worked in spinal cord injury, let's try another indication and see if it works. And we apply the same things after optic nerve crush, and we see a positive ERG. A healthy or damaged eyes after treatment with ExoPTEN response to light in a dark adapted experiments on the same way, time-wise and amplitude. They are aligned. The graphs are aligned after 18 days, meaning that there is the same response to light, okay, after using ExoPTEN. And then we have also sponsored research dealing with facial nerves. Also there we can really see results, nice results, dealing with, facial nerves is semi-central nerve system. It's not 100% central nerve. It's a bit more peripheral. But also here we can see an improvement. So with that, we're taking all the material that we produce into this. It's part of our IND enabling study, IND in investigation of new drug, and this is what we want to submit in the coming months in order to start in the clinical stage and provide it to human.
Awesome. And in that the rat spinal cord injury study, that was you had control, right? You had a placebo as well.
Yes, of course. So we use exosome, for example, as our control, for example, to see if there is effect of exosomes. And there is some effect of exosomes. But as I said, because there is a therapeutic effect of exosomes by nature, this is there. I mean, you have exosomes in your body exactly for these reasons, to communicate with healthy tissue to damaged tissue. But in spinal cord injury is very tricky because the threshold, you need to pass a threshold which is very, very high. So you see some effect, but it doesn't go to the extensive effect that we see with combining exosomes and the siRNA. So we see synergistic effect between the two components, the exosomes and the siRNA.
Interesting. And with again with the spinal cord injury, can you talk about more? Did you do like you still did like a histological analysis on the spinal cord afterwards? Did you
Yes, of course.
what did that look like?
So we have several levels. So we have a MRI that we did, and we use a DAT in order to understand the vector of the water, how the water flows in the spinal cord injury, in order to show the right connectivity. Okay, this is one. Second, we have a normal slides for histology. Okay, Some case we also measure we use specific antibody for vascularization, for example, to see if the blood vessels are different. And we do see that after the treatment, you have a larger blood vessels in the area, meaning you have more blood flow, more cells could be survive or regenerate. Okay, so there's another indication. So different slides is also relevant. Yeah, that's mainly the things that we do after, the physical measurements, which cover PPP. In the last experience, we also had catwalk to really analyze the walking pattern. And yeah, so that's what we do.
Awesome. Yeah, I've seen the videos of the catwalk. I mean, it's pretty
Yeah.
clear as day.
You really can see that they walk, and you really can see, when you deal with a compression model, there are physiological models that are very, very tough doing on rodents, okay? Because you have self-recovery, okay? So the fact that you are compressed or impact is the second one, impaction. If you impact or compress the spinal cord injury in rodents, rat could recover even without treatment, okay? And you can see that there is some movement also in the control, okay? But in order to really to differentiate what the exosome SPT10 group and the control, you need a very dedicated machine, which is catwalk. In this case, the fact that it moves or get a high BBB score, BBB is the motor score, okay, which is very subjective because you as a scientist sit in front of the rat and say, he moved, so I tick the box. But when he moves like that, but will not walk, what does it mean, What does it mean? So although the BBB is getting higher and higher, we want to understand that we actually can promote walking and movement, right movement for those rats.
Interesting. What I kind of want to touch on some more things with exosomes because they're getting pretty big. And you mentioned years ago that people came out and kind of discovered that it's not always the stem cell itself; it's what they secrete. And I think Dr. Arnold Kaplan, one of the godfathers of mesenchymal stem cells, came out with a paper like 5 or 6 years ago and said this basically that wants to change it to medicinal signaling cells, and they have a lot of advantages. Some of them we've already talked about, but some we haven't. So size, for instance, which makes a big difference for a lot of things, potentially blood-brain barrier, but also pulmonary first pass, um, and ease of administration and logistics, handling those things. Could you talk more about some of the advantages of exosomes over, live stem cell therapy?
No, sure. First of all, I would say I'm coming from the liposomes area. Liposomes is the artificial drug delivery systems around 108, I would say 80 to 100 nanometers. So you're talking about nanomedicine. So it's in a nano scale, talking about 10 in minus 9 size. So they're very similar in size to liposomes, but not like liposomes. They are passing the BBB, the brain-blood barrier. So for us, it's a huge advantage when we are dealing. Our company is focused on central nervous system. So we need a drug delivery system that can work. Second, they have, as we discussed before, therapeutic effect. They have therapeutic effect. So it is not just drug delivery system, it's a drug delivery system with a therapeutic effect. Third, we are talking about allogeneic approach. If in the past the cell therapy were, stuck in the conception of let's have cells from you as a patient and we do a precision medicine, we don't need you will be the donor of yourself and we take your blood, you take your cells, engineer it and return it to you. Now we are talking about cell therapy without cell. I can use one exosome batch to treat several patients. There is no immunogenic activity. And think about it, it makes sense because if I'm going to donate blood to you, what are the questions that I ask? my cell type blood, what is the blood type, okay? AB on O, and plus minus. Do you think that I'm not going to transfer you exosomes? Of course I am. Does it matter? No. And this is exactly what we see. It doesn't matter if you treat a rat with a human bone marrow derived exosomes, okay? All the experience that I discussed before were done from a human source on a rodent, okay? Exactly here with the human, you use one donor for human for several patients. So this is a huge advantage. So being in the allogeneic space, make it off-the-shelf product, you know, not out, it's not over the counter, is that off the shelf, meaning that I can treat with one batch several patients. What else? I think we didn't touch it, but a bit, we said it a bit, but when you are entering to the nanoscale medicine, you don't deal only with the therapeutic effect of the single molecule. You are talking about the physicality of the program of these exosomes. If in liposomes, one of the effects that create accumulation is the EPR effect, enhanced permeability and retention. Here we are talking about the effect to see accumulation of exosomes from the fact that the exosomes themselves are homing to inflammation. So think about it. You have a tiny missiles, guided missiles to inflammation without the need to engineer them. This is the huge advantage with exosomes. And from my point of view, so far, as much as I worked in the exosomes close to 5 years now, I do things that exosomes for our indications consider to be the ultimate drug delivery system.
Yeah, I mean, there's a ton of advantages there. There's also, even from a business and like logistics perspective, I've had multiple bone marrow aspirations myself, and they are not fun. And if you're doing something like that, you have to go in that day under anesthesia, get it done in the process, and then if your procedure requires another anesthesia, it's a long day. So to have an off-the-shelf product would be, if it works, it'd be a pretty big breakthrough.
And this is that you said something, another thing. You took it to to another thing that I wanted to mention. I'm taking it for the operation again. Beside the fact that it's off the shelf, when you move from a cell therapy to exosome therapy, we are talking about not having a huge fermenter. Okay, the gold is not the cells. The gold is the medium. Okay. Now, when you think about it, you have, in order to have like a large scale manufacturing site, you're not talking about 20,000 liter of bioreactor. You're talking about liters of bioreactors. The cells are there, stuck there, and you perfuse medium again and again in order to milk the cells from their exosomes. So operation-wise, you're talking about tiny bioreactors that can produce a huge value. And this is another positive point for the exosomes.
Yeah, yeah, and I'm sure at scale it would be much easier to do that than do it for each individual patient, and easier for the patient too.
Yes.
And then transporting these things. So if you want to transport like a mesenchymal stem cell therapy or what have you, you have to go down to what, like 80 degrees Celsius to freeze them. Minus,
Yeah.
yes, of course. And exosomes, how cold do you have to keep those for transporting?
So disadvantage of exosomes is their stability to a freeze-thaw cycles. Okay, freeze-thaws are not so good at that. So they are very okay with minus 80, and they can also survive, of course, in minus 20 for a shorter time. But there are already companies, and this is not our technology, that you can actually prolong the shelf life by lyophilize the exosomes. So you actually get a powder and then redistribute it with the medium to injection. So at the moment, we have the -80, and this is just to get it. Our intention is to get to clinical stage as fast as we can. Okay, we want to show clinical efficacy and that it's not toxic. When we are dealing to a longer shelf life, this is of course under our radar, and it's possible. It's not part of our added value. Our added value in the patents are dealing with the production, the loading technique, and what we load. And I believe with the time, maybe a second generation lyophilized will be also a part of Nurexone. Yeah.
Fascinating. What are those? So next steps is phase 1 in 2026, you're hoping?
We are hoping to submit in the second half the IND, and then open several centers in the US, Israel, maybe also Canada, for this study in spinal cord injury. There is also inspiration depend on fundraising to do it also in glaucoma or optic nerve, which will be faster, but it's not orphan drug designation. And everything is dependent on the fundraising, in this situation, because when we are dealing with orphan drug designation, you need to make sure that you complete it fast. So you need to do several centers, and those centers should be large. So it's like a large trauma center for a very large population with the rehabilitation unit, because it's also very important for spinal cord injury. So those are the things that we map at the moment, and we're looking forward to work with the right CRO for that, to get the study done.
That's awesome. So the trial will include rehabilitation as well.
Yes. One of
Okay.
the key point that we want to measure during the first phase 1/2A is, can we actually reduce the time of the patient during the trauma, the trauma center? So meaning, can we actually move due to the treatment, can we move this patient faster to rehabilitation? And if this is right, then the commercial aspects of the ExoPTEN is even beyond what we do for the patient. There is a financial aspects for that for the hospital. The hospital is a motivation is to use this drug in order to clear a bed so we can have another patient.
Yeah. So you mean short? So will that be an
Shorten
endpoint?
the trauma center. Yeah.
Shorten the time from trauma center to rehab, and that will be something you'll measure,
We
obviously.
would like to measure it and see if we can have any commercial aspects on that for the hospital.
That's really interesting. Any other interesting endpoints or things you'll be measuring during these trials?
Of course, sensoric aspects. We are going to take, of course, as required, different biomarkers. This is also the reason that we move from intranasal to intrathecals, because anyhow you need to go intrathecal to take some biomarkers. We also going to see who is the decision makers. When you are entering to as a clinical patient to a hospital, who will give you the drug? Is the neurosurgery? Is the orthopedic, because he needs to do operator, because you get your spine inside, crash into the spinal cord, so you need also operation. This is always will be part of among the standard of care, yes? So who will get the decisions? What is the effect of the rehabilitation? We discussed how if we move it faster, but what is the protocol for the rehabilitation, what we put inside as part of the treatment, because it will be ExoPTEN plus rehabilitation in the end, like we do with rats. It's not standalone. Yeah, those are the questions that we are dealing with.
Interesting. I know that there was one, I think it was a Wharton's jelly intrathecal study. I can't remember where, a few years ago, that seemed to do okay. And then there's one that just started, I believe, in Spain at the Gutmann Neurorehabilitation Hospital. They're doing intrathecal orange jelly plus rehab. And yeah, I guess we got only a couple of people working on this, but it'd be really interesting to compare your results to whatever comes out of the Spanish study as well using
There is
cells.
a Spanish study. We have also a Canadian company that trying to make it in the
Mm,
spinal cord called Nervegen, the clinical stage company. They just published in Asia the results from the first phase. They could make it to the primary goal, but not the secondary. So there is question here if they will make it to in the phase two. Yeah, I do believe that it's good to have competition. Uh, this space needs revolution, needs innovation. It's at the moment really blue ocean. A lot of company, or some case, if you ask VCs, it's a graveyard, and we need to have a change. I mean, we have AI now. We have such a lot of innovation in different aspects, but so far we couldn't make it to regenerate nerves in the spinal cord after spinal cord injury. So if we will make it, you know, that would be amazing. It will be amazing.
yeah, and I think all that really matters is that there are lots and lots of people out there suffering who have a tough life and no answers, and it's not a zero-sum game. So the more, the better, and the more data we get, the better. I think you guys are doing it right. So
Exactly. And this is what I'm trying to explain to a lot of patients in suffering from spinal cord injury. Our drug, Nurexone drugs, is for the acute phase. I will not be able to treat them at this point. It's a
mm,
bit too late. But let's think about the world that my drug was in the market when you were injured. Okay. Maybe your life would be different. Okay. So help us to make it happen. Okay. I do hope that after positive results in the acute phase, we will be able to, combine it with a cell therapy, because it's a different. I mean, if you are chronic spinal cord injury, you need more than the exosomes. You need probably a cell therapy plus exosomes. And this is what we try to do with a company called Intelligix from Canada, a company coming out from Professor Felling lab. We try to also address spinal cord injury patients um, in a chronic phase in order to give them also hope. That's really important. Yeah.
yeah, totally. Um, cool. Anything else you want to run through? Anything else that we missed on the company or you or the products?
We can speak about it. Why the heck we decided to go public so early? This is another issue that we can talk. It's
Yeah,
maybe less science, but I think I find it very fascinating because our world is changing, and it's very, hard to secure funds, uh, especially in the field which is considered to be graveyard. So what you do when you have innovation, and I believe your audience, some of them are entrepreneur scientists, would like to make their technology into a company and try to help patients. So uh, one of the reasons that I choose to become the CEO of Nurexone is, of course, the science behind it, and I felt comfortable taking it ahead. But the second one is I really wanted to show by my two hands that it's possible to have an alternative way to finance biotech. And when I say biotech, it's the most extreme biotech. We're talking about biopharmaceutical company that get the product into the market between 10 to 12 years. So how you will manage to recruit, to fundraise after fundraise after fundraise, and you don't have revenue. Um, so I saw cases that, VC were very, tough on the companies. They took too much control and they actually killed the innovation because nobody wants to invest in a company that was actually taken by VC as a majority. And I'm talking about even 80% of the company after first round. it's kill innovation, and we must have another alternative. I'm not saying that what we did is the right for all companies, but I do think that there is alternative ways to get the innovation out and say and shout loud, here we have something else, and try to call the other investors, other people around, listen to us. And we are very tiny, listen to us. We can make it happen, invest in us. And when you are transparent, when you're working about a lot of IR and PR to get your message across, you can see a success. And we are in the middle, we are not done yet. Okay. We need to get this company into the real market, into the US, New York Stock Exchange, NASDAQ. We are still in Canada. Canada is not a favorite environment for the US investors. And this is what we do at the moment. I'm very happy that at least during the phase of being a Canadian company, we could mature. We can work as a publicly traded company, release the MD&A, the financial report, working in the highest standards of regulation. So you gain knowledge as a publicly traded company. And second, I would say we could see that what we do so far, at least in the Canadian, works, because last April I received an email and say, we invite you to come to Vancouver because your small company called Nurexone, after 2 years, because we just made the RTO reverse takeover, become public in June 2022. After 2 years and a bit, I was nominated to be part of the best 50 companies of the exchange. And in this exchange, just to let you know, there are more than 1,600 companies. And we are position 49, part of the best 50 companies. And this is regardless on the scope. They took all the pool and asked the questions: who made it the share appreciation the most, liquidity, and other economical indication in order to make the list. And I'm very happy that we are part of it. I think you tick the box also for that. And I hope we can make it also nicely in 2025. But in the end, our product is the science, and our product is the team. We have a very, very good team of scientists and people in the company that knows how to execute. And if you have a good story and you can execute, which is important, investors comes.
Yeah, absolutely. And honestly, I think you did the right thing by showing off the rat model videos. That's pretty, like I said, that's pretty telling. So I imagine that helps.
Thank
Yeah.
you. Yeah, it is. It's very visual. That's true. But it's good for investors when you show it to a spinal cord injury ventures. They said, okay, do you try to check other things than motor? Because this is the thing that they check you. And I say, yeah, of course. Because when you deal with spinal cord injury, as I mentioned before, motor recovery is not the most important. It's very visual. It's caught the eye. But when you want to get a non-dilutive grants, you need to show much more than that.
Sure. Yeah, I mean, I imagine getting in the door, and then you could, I mean, you know this better than I do, but have a discussion about, if we could prove that this regenerates the big nerve, the spinal cord, which you're going for because you got the Orphan Drug Designation, because there's not a lot of answers for. Then once that's sort of proven, there's all kinds of peripheral neuropathies and all kinds of
Exactly.
neuropathies. So
That's it. That's the idea why to continue working with spinal cord injury, get the support from the FDA. We receive also very detailed response from the pre-IND, really helping us to map what is important, what is not important for the FDA in order to submit the IND for spinal cord injury. Communication with the agency is very important. And the fact that we got the Orphan Drug Designation will allow us while submitting the IND, also to have the fast track and also maybe the breakthrough technology pass in order to get the full support with the development of this drug.
Wow. How has I guess last question just curious, how has working with the FDA been? It's been a crazy year. I don't know if you saw last night, Dr. Prasad left CBER and amongst other stuff. So how has that been this year for you? Has been still fine?
So the last communication was before the changing over the administration. It's a not easy situation. I do understand it. I do think that US is the main market and we need to continue with the FDA. The results are there. There are other agencies to work with. We get a lot of attraction from the Far East, actually, South Korea, Japan. There's also very, very advanced market in the regenerative medicine. If the science is there and there is results, we will find a way to get it to the US, and the FDA in the end will get the results and hopefully will approve it. We still need to deliver the preclinical package. Synopsis, I think we are in a good shape. How our clinical phase will be. And also we are quite advanced already with the preclinical data. We have the toxicity to do, the CMC needs to be done, the spec. So this is where we are. So in the middle of the process.
Wow. Cool. Well, those are all the questions I have for you. I hope that we can, one day look ahead in 5 years and say, remember that time we sat down and talked about this and you guys were just in the very middle of developing all this stuff and now look at you. So really interesting interview and appreciate your time.
Thank you so much and keep innovate, I would say. Thank you.